Let me start the course. So as I mentioned, we will have an active learning process in which you will involve how we develop the material. You will ask active question, okay? And I already invite some of you. If you are not invited yet to my curve note, so let me know. So let me check. So Christian is already here. You can open your curve note, go to project share project folder, and you will find this book. If not, let me know. And any more, Majere, you also invited. Jonathan, you also invited. Trianto, you also invited. Padin and Sinta and Edwin is also invited. Okay? So let me tell you the motivation of this course. So with rapid development in artificial intelligence, the way we learn bioinformatics or any science should be changed. Initially, we learn by reading textbook, and we have hard problem on implementing algorithm that we learn from mathematical textbook or bioinformatics textbook. Yeah. So with AI, you no longer have that problem. Okay. All the code in this book is generated by AI. AI have more better capability in translating mathematical concept into pseudocode or into any language. You can ask the language in Python, you can ask the language in Rust, but in this book that we will develop together, you will be part of the author list and you will take this opportunity as a part of this course to contribute. Yeah, I will lead the development of the book, but I will need some help from you for proofreading, give more comments. If you think you don't understand, give the comments. Yeah. You can also participate in helping the code working well without bugs, because sometimes AI like GPT or DeepSeek or Cloud, okay, still have hallucination on code. Yeah. So we will need help for you to do QA, quality assurance on the code. AI that we will use, of course, the first one is ChatGPT, this one. The book will be generated by O1 and O3. Okay. This is a very powerful AI. It can write any codes you want. Okay. But if you don't have the paid subscription of O1, you can try DeepSeek. Okay, you can start, open it, and you can also have limited token in cloud. Okay, it's very useful if you want to debug or bug fixing any code. So we will use these three AI together to develop this book. And why Rust? Actually, any Python code, you can ask to ChatGPT if you understand what you want to ask. The problem with Python is we are dealing with massive data in bioinformatics. And if Python is not capable, we need to be able to have option. And the option should be lower level language like C++ or Rust. But many community now, especially in computer science and bioinformatics, is evaluating Rust for alternative language alongside with Nextflow. So Nextflow is workflow engine. Like if you want to do computation in terms of scientific workflow like bioinformatics, then you can implement the logic of your workflow using Nextflow and you can execute. But each logic you can implement in Python or you can implement in any language like Rust. In this course, we will use Rust, okay? But anytime you want to transfer the code to Python, you can do it as well. Okay, you can do with the help of AI. So before we start, so please download the Rust. Go to Rust programming language. Maybe teaching assistant can help to give the link into the chat room. And then you can install. Okay. Just download this, copy this and running in your command prompt. And also you can go to Nextflow and install Nextflow. Yeah. You have to install Java first. And after that, you check the Java and install using this link. Yeah. I already installed in my machine. So what happened after you install? Okay. So first make your working directory. I would say let's make BGVR working directory. Okay. And then I go to BGVR. Nothing there. I just created. Okay. Once you install Rust successfully, you will have this program called Rust-up. Yeah. You can say Rust-up minus V. This is the version of Rust I'm using. If I want to update my Rust, I just say Rust-up update. Okay. But I believe this is the last version. This is the whole command. You don't have to memorize. Now it's downloading new version. Yeah. So you can also download now and try to, once you have download and installed the Rust, you will have Rust-up program. Yeah. Okay, other thing that you have to download is your IDE. I like to use Cursor, not Curvenode, Cursor, because it's well integrated with AI. Okay, try to download this IDE so you will have installed in your machine. So here I'm still installing Rust-up. Please download. Okay, Rust-up is there. So I can check again Rust-up minus V. My version is 1.27.1. Okay. And after that, you can download your editor and you will have editor like this. Because I create my working folder in BGVR folder. So I will choose this folder as my working directory in my cursor. Okay. And you can open terminal in your cursor. Make sure you are in the folder. Okay, I'm ready. I can call Rust-up. Okay, I can get the Rust-up program here. If you already install Nextflow, which is not necessary for this time, but try to install, you will have Nextflow. Okay, this is Nextflow. Yeah. Okay, so all set in my side. While I'm describing the material, you can start install Cursor, Rust, and Nextflow. Okay? Okay, let me continue. So this is the motivation of the book that I wrote for you. Yeah. So we want to have a book that we're happy with it, cover the fundamental, conceptual, and practical aspect of how to understand bioinformatics and functional genomics. And this book is also powered by AI, so you can always get knowledge from AI, get the code from AI, and you are the one who will complete the book with me. Okay? So I suggest you put your name here. I will give the example. Also, our teaching assistant, Sinta, Fadin, Edwin, give the profile here. How to edit, just edit. Okay? How to make my profile, for example. Yeah? So I just make like bachelor degree in physics, Universitas Indonesia. Okay? Master degree in physics, Universitas. You can give the year. I graduated, if I'm not mistaken, it's long time back. And PhD, a doctoral degree in computer science, uh, Universitas Indonesia. I'm advisor, industry advisor from of Data Science Center, Universitas Indonesia. Okay. Okay. So once I have the bullet points, I go to AI. I just say, wait, write short bio about me for a book called BGVR, uh, Bioinformatics and Functional Genomics via class. Okay, I just give the okay. I like this one. If you don't like, you can ask again to change. Let it wrote by your instruction, and I will put here. I will ask you to do the same. Yeah, and put just one paragraph like this, and then save. Okay. So put all of your name here, just one paragraph, simple like this. Uh, Everything will be okay after that. Okay. So you are here. Yeah. And we need to have Profile Hadi also, as he will be the final reviewer of the book. So let me share with you the plan. So we plan to deliver 25 chapter in 14 weeks. It's almost impossible before that you can make a book that cover fundamental, conceptual, practical. But in AI era, that's possible. Okay. So we have four parts of the book. The first is foundation. In this case, we will introduce what is bioinformatics and functional genomics, setting up your development environment, including RAS and Nextflow, key data structure in bioinformatics, key data structure and algorithm in genomics, core bioinformatic tasks like sequence analysis, high throughput sequencing, genomic data parsing, variant analysis, And all functional genomic common case like gene expression, epigenomics, metagenomics is microbiome analysis, and population genomics, and some of Nextflow core, NF core reusable pipeline, integration with AI, okay, including reinforcement learning, machine learning, deep learning, and large language model, yeah. Uh, and final one, we will also cover future direction and emerging trends, okay. So I already wrote for you this part, okay. You can read. If you want to change anything in this place, just put comment here. Like give guidance to blah, blah, blah. You just put comment here. Okay. After you read, put comment. Okay. Or maybe you want to edit something. Let's say this part, cover machine learning, deep learning, reinforcement learning, and large language model, right? But this part is not translating the idea. So I can say here, advanced AI application. Okay. Okay. And if something here need to change below, it just change below. Okay. I need you to proofread. Make sure that we didn't make any mistake in this page. Okay. Preface, we will make it later. Forward, we will make later. This is also later. Let's start with chapter 1 for this, for today. Okay. I already wrote it for you. Please read. Yeah. And if you don't understand, please comment. Add comment here. Okay, like I need more explanation about this. I need more explanation about this. We will compile your feedback and make it adjustable with your current understanding of what is bioinformatics and functional genomics. Okay, each chapter we start by quoting some relevant Nobel Prize or top scientists, what they say about this chapter. So let me start by Leroy Hood. Yeah, he said biology has become an informational science, and it is increasingly clear that integrating computational and mathematical principle will be essential for the next generation breakthrough. I love this. That's why it's here. So the first section is about what is bioinformatics and functional genomics. Yeah. So we have to look on the history. We have to look on the future trends and what is in between. We cannot just introduce from decades ago or from future, but we have to have a robust introduction. And this is what I came from. So bioinformatics started maybe in early 1960. Yeah. So this is actually a academic term for how we can process biological data or biotic data using computational and statistical techniques. Yeah. And then there is also improvement in terms of the bioinformatics from Paulen Hogeweg in 1978 in context of informatic processing. So it's combination, we can say it's a combination between computer science, statistics, mathematics, and maybe physics, because we are dealing with sequencing device that working with principle of physics. So it's an intersection between biology, computer science, statistics, and more data science or related things. Okay. If you want to put data science stuff here, just put in comment and I will put in the text. Okay. Okay. So all bioinformaticians start by learning the central dogma or biology. So what is this? There's a lot of story about this from biological sides, from bioinformatics sides, yeah. So this figure is actually describe the central dogma that genetic information inside a cell, there is a chromosome. Inside the chromosome, there is a long strand of DNA. This DNA, yeah, uh, will be transcribed by the RNA. Okay, so the segment of the DNA copied the into the messenger RNA, yeah, is kind of like a blueprint in our biology. Okay, this blueprint is like cellular machinery that generate the protein in our body, yeah, and this becoming the blueprint of everything happen in our body. This is the dogma. Okay, so the DNA itself contain nucleotides labeled as E, adenine, thymine, guanine, and cytosine. Okay, so we call it ATGC. You can imagine this is a basic building block of double helix, ATGC, yeah, paired together, very long strand. Even if this is, you can make a string out of DNA, you can go to Jupiter and go back to Earth if you take the string, yeah. So it's very long and very dense information inside, but the basic building block of DNA is actually a TGC. Okay, you don't have to memorize this. Everything is everywhere in the paper, in the internet. But to make this book better, we also have some referral, yeah, uh, that we generate. So I also need help from you. Once we fix the chapter, okay, when we fix, you have to find out which one of these reference is still valid or not exists, yeah, because sometimes the AI is hallucinating regarding reference, okay? But we need to proofread with our common sense to make sure that this book we're happy with to learn with, and it is the best that we can come up, okay? So another concept that you have to understand is not only the nucleotides inside the DNA, like ATCG, but it's also the SNP, yeah? We call it single nucleotide polymorphism or SNP. So it's a single base change in genomic sequence, yeah? So we can illustrate this, like three human being here, color code with different color code, yeah? So maybe they have a sequence, but there is a variation or there is a polymorphism between these three persons that at the fourth element of their sequence is becoming different. Like person one get A, person two get G, person three get T. Yeah. So it's not a major switch. Yeah. But a small polymorphism inside the different three different people of this one. Okay. But the individual genetic usually have a long strand of this DNA, yeah, but each individual maybe have difference in terms of their SNP. Okay, if you like this explanation, just let this like this. But if you think we need to change, you have to suggest by adding some comment here. Okay, I continue. How we can get the data? Okay, usually we take the sample of our blood or saliva and then go through sequencing devices. Modern sequencing is high throughput sequencing. Okay, if you want to add more information about another device, you can just put comment, explain this before this. Yeah, but this is what I bring to you. Like I want to describe directly into a high throughput sequencing because it's more modern and sequencing platform like Illumina or Oxford Nanophore or PacBio. Yeah, we also discuss after this. But this is how will be the sample will be processed. So from the sample, like your saliva or your blood, it will be prepared for isolation to make sure that this isolation can isolate your cell at the DNA or RNA level. It's quite different isolation technique between DNA and RNA, and it will prepare the library. So this library is like a rack before you read it, yeah, and it put into the devices and then start the sequencing process. There is a data analysis process usually embedded in the sequencing device, okay, that we sometimes call primary analysis. Okay, the workflow is described in this paragraph. Okay, what is the output of this device? We call it FASTQ. Yeah, if already have binary alignment file, we call it BAM. There is many type that we will explain later in different section. Okay, so if you think it's good enough, let it like that. But if you think we need to put more, you can do like this, like explain more about blah blah here. Okay, you can put here, or to get my attention, you can give different color. Okay, how to give different color here? Wait, or bold, yeah, to get my attention. Or you can put in the command. After you put that, you can save the version. The curve node will save the version, so I can go back to any version I want. Okay, don't worry about mistake or something else. Okay, this is the first section, and then I describe functional genomic at the introductory level, like is like a subset of bioinformatics that focus on characterizing the roles and interaction of gene and the products developed in tandem with bioinformatics. Yeah. So usually, by functional genomic centered on analyzing gene, okay, and phenotypes. Okay. If you don't understand phenotypes, you can comment, yeah, like usual, you can comment. Make sure that you can differentiate between functional genomic and bioinformatics. Okay. So technology moving from 1970 up to now, the last decade, people already know technology like CRISPR. This is the gene editing that not only read the patterns or discover the structure inside your gene, but it's also we can edit the gene, manipulate the gene. Yeah. So this technology invented before, and the founder get Nobel Prize. If you think we need to put more information about CRISPR editing, you can also mention. And I start to think to mention here that after the bioinformatics, and then there's a statistical physics where some idea is actually uh, we got we borrow from statistical physics. Let's say Markov random fields. So MRF is usually how we read the data, interpret the data from the device. Right? This picture is beautiful from a paper called Grace. Yeah, from transcriptional data set, and then it creates some binding motif filtering, like gene regulated related information, and then it build the network, and then create another meta network, okay, and then perform some another analysis and ensemble Markov random field. Okay, this is just illustration how some idea actually borrow from physics, because this kind of process in physics is very common. Okay, from computer science, usually we describe the structure of gene in terms of graph. Okay, and common graph that is used in bioinformatics is the Bruegel graph. Okay, so this after the read, yeah, we have to build a graph. We call it the Bruegel graph, yeah, that read the sequencing and structure it inside the Bruegel graph. We will explain more in later chapter about data structure, about the Bruegel graph, because this is just introduction. We make it dense so people can get the basic idea first. And then people start use neural network for computational, graph neural network, and any other topic like pangenomic, in which we address the observation of single linear reference genome. Okay, we call it pangenomic. It's also using graph like the Bruegel, yeah, but it's different kind of analysis. We will also explain this in later chapter. And some people start to utilize deep learning, okay, artificial intelligence. And after that, I always give sample code in RAS, yeah. So for example, this sample code, read uh, how to build MRF from FASTA sequence, okay, from the sequencing. This is the sample code, yeah. I will make sure that the code working well, but I need help you to read the material, proofreading, give a comment, and later with teaching assistant, I will make sure that the code working well and organized well. Yeah, this is not organized well yet, but will be. Yeah, and we will explain bottom-up approach. Make sure that you can regenerate the code. Okay, you can reproduce the code. You can make the code working well. If you don't like using RAS, you can simply translate the code. For example, I don't like this one, so I will copy here, and I will say to convert the code to Python and use related command library in Python. So code translation, one of the best capability of AI. Okay. So it's also have a bio, actually. RAS bio is libraries coming from Python, convert from Python, and you can get this, yeah? Once you get the Python code, you can run this here maybe. You have to install the library. Yeah, still error, okay? We will bug fixing later. But I just want to show there is no issue translating code from one language to another language. We will much focus on RAS in this book. The reason is there is no book in RAS for bioinformatics in the market yet. Okay, so we may be the first. Okay. And especially in combination with Nextflow. Okay, that is the first section. And next, I will cover the second section. Okay. Here we will explain more about mathematical concept. Okay. So sometimes the prompt that I use to generate this content is too much redundant. Yeah. So if you think redundant, you can give command or revise directly. Okay. But in this section, I explain what is Markov field. It's coming from physics. So some distribution is usually from statistical physics, especially explain that this kind of a potential, yeah, pairwise potential, very common in statistical physics. Okay, but also coming from information theory like Kullback-Leibler divergence. Yeah. So this mathematical, you also have to read. If you don't understand, give command to make sure that we can come up with the uh, good enough section. Yeah. So of course, chapter 1 will be hard because it's a summary of all chapter. Yeah. Uh, Everything else will be easier after you go into the next chapter. We cover more specific topic about data structure, but this chapter 1 usually very dense. Yeah. But will be explained more deeper in following chapters. Okay. Data and Computational Foundation, section 1.3. I'll cover about the type of data. Usually you have to understand many types like FASTQ, FASTA. Yeah. I miss FASTA, so I have to put comment here. Explain also about FASTA file. Okay. I just explained FASTQ. Yeah. So this is the first raw format you get from the machine. And then once you have sequence alignment, you will have different file format, SUM or BUM. Yeah. I try to give the sample code, the sample file uh, here and explain the history. And I want to explain how you can use RAS to download data from non-URL in NCBI or other source. If they're using FTP or HTTP, we have to be able to code in RAS to download the FASTA file. Okay. Next, this is all mathematics. Explain the background. Yeah. We don't have to memorize the mathematics, but we have to understand at least the complexity of the algorithm, the correctness of the mathematics, okay, common methods. So you can utilize AI to generate code, whatever you want. If you don't understand the mathematics and the algorithm, hard for you to generate any code that is correct. Okay. And usually I also give sample code for implementation and explain the code. Tools and framework, we will use some basic tools from bioinformatics like genomic readers, visualization like igv.org. We will use Nextflow. Nextflow is a domain-specific language for scientific workflow. It's okay if you just heard it. Before Nextflow, people using many workflow framework like SnakeMake or CWL or many things. But looking on the industry now, they want to have fair principle in place like findable, accessible, interoperable, and reusable. So Nextflow is the best to implement this principle. So any workflow and pipeline from bioinformatics can be reproduced so anyone can improve in open and honest circumstances. Okay. So the bioinformatic computation should be findable, accessible, interoperable, and reusable. So we will use Nextflow. So this is why we introduce Nextflow. Nextflow is language is using another language called Groovy. Don't worry about this language because mostly AI will help you to write if you have a good prompt. Okay. Most of the code is generated by your understanding or your common sense. So we will give example that works. Okay. And this is another sample code that using Nextflow and also RAS. Yeah. And conclusion. Okay. So I will taking care the full material writing based on my common knowledge, my understanding, and to make sure that you also learn actively. I want you to read, and if you don't understand, put comments. Yeah. And how you can learn more is I provide many prompts here. Like if you want to know about MRF, just copy this prompt and then pass to your AI. It will explain me more deeper. But of course, it's hard to put every content inside the book. Yeah. But if you find something that you think we need to put in the main section, put in the comment. Okay. And I will revise the material. Okay. So you can see here, that's just one of 20 prompts. Let's say I want to understand about single cell RNA. Okay. Wait. I pass this. Or maybe you can modify before you pass. You can see now, right? So if you think any new concept should be in the main section, let me know by putting comments. What else? I give 20 prompts here. Yeah. The last section, always problem solving assignment. You can do it manually, but I know it's hard. But what you can do, you copy this and then go to AI and then say based on latest state of the art techniques, work and solve this problem. And based on your common sense of what you learn in the chapter, you can assess, okay, the preparation, data acquisition, quality control, normalization, okay, choose MRF types, choose factorization, okay. See, give you more detail, okay. You can also mention, translate the code in RAS. So language barrier is no longer exists in the era of AI. I don't want you to learn something and tie it into specific language. So if you want to develop products in the future or service in the future, you can implement in any language. If the products need low level or machine adaptation, you can do in language like Rust. But if it enough, you do it in Python, you can just do it in Python. Okay? Okay, good. So that's all for chapter 1. Okay. Because we only have 1 hour per week, I suggest you self-read chapter 2, give comment before I finalize. Yeah. What I will do in finalization, I will make sure the code is running well and your feedback is accommodated. Okay. As a preparation, you can install Rust and you can start learning Rust together with Python. I believe you already know Python. Yeah. So I assume you already know Python. So I don't teach Python anymore. I just want to let you know that any language is no longer different, but we need to pick the language that match to what we will require in the future. With Rust, you can parallelize the code easily. You can deploy the code in high performance computing easily. Yeah. And you will learn that in chapter 2. Okay. So I will stop now. Next week, our focus is to complete chapter 2. Up to chapter 2, yes. So you have to prepare your feedback. You have to install Nextflow. You have to install Rust. You have to install your IDE, Cursor IDE. Yeah. Next week, I will cover data structure and algorithm for bioinformatics and genomics. Okay. So every week, I hope our speed is 2 chapters per week. Okay. Before I close this lecture, let me know if you have any question. Okay, there is a good book that we wrote before, PRPL. Okay, this is for Rust language. And another book for data structure and algorithm. I assume all mathematics students already have data structure and algorithm, but we make a book for data structure and algorithm on Rust that can help you to up and run quickly. Yeah. The same style with the book that we are writing. In every chapter, you will have prompts. You know how to generate the code. You know the fundamental and conceptual of each topic in the chapter. Okay. So take a look on these 2 books for preparation, and your homework is to reread, proofread, comment about chapter 1 and chapter 2. Okay? Okay, so if no question, I stop here. Do your homework, read chapter 1, chapter 2 in Kernel. Give comments, yeah. Wait until the code is finalized by me. And later I will need help like mathematical notation or glossary, yeah. But for now, just proofread and give comment. So any question before I close? Trianto, Majere, Christiane, Jonathan, or you need time to digest or to install everything to prepare?
Yes, sir. We are starting install.
Okay. Start install your cursor, your RAS, your Nextflow. If you need guidance how to run Nextflow in browser, you can ask to Fadin or Sinta. We have the guidance because not all machine is capable enough, I believe. But I will use my local PC for writing the books, and I'm using Mac. But if you are using Windows, it's okay. You can try. Okay. Make sure you can do Hello World in RAS, Hello World in Nextflow. Yeah. Using Cursor IDE. By the way, Cursor also integrated with AI. So let's say you have a code, you can just ask how to write the code from the cursor itself. Okay, we will show you later next week. Your homework this week: prepare your environment, Rust language environment. Read chapter 1 and chapter 2. Next week we will cover more deeper aspect of data structure and algorithm for bioinformatics and genomic in chapter 3 and chapter 4. Okay. So thank you, everyone. So see you again next week. Spend enough time in this course. I know it's not easy, but I believe the journey will be beneficial for all of you. You will learn how to make books, which is good for your master degree thesis later on. You will know how to make, let's say, working with AI, which will become your assistant when you are doing your research later. So all the benefit will go back to you if you spend enough time. At least in a week, you spend like 5 to 10 hours. That will be enough. Okay, thank you very much and see you everyone and next week.
Terima kasih, Pak.
Thank you, Ustaz.
See ya.
Thank you everyone. Terima kasih.
Thank you.